Thank you for being with me again in the fourth week of this NPTEL course, Functional Genomics. In the last 3 weeks, we have been looking at how technologies have changed, how we have understood the genome, how we have understood the transcriptome, the expression profile, and how we can engineer the genome and so on. So, in the last this lecture and a few more lectures that we are going to look at is to sort of go back and look at our evolution as to how the genome has changed over millions of years as we evolved from simpler microbes to multicellular organisms that are invertebrates and two vertebrates like through fish, frog and mammals, how our genome has evolved and how the complexity has come in and how the information that we can go back and look at the genome of diverse organism can help us to understand the uniqueness of certain species, including humans, and that would contribute in our understanding of disease or to making better animals that are domesticated, to identify even the evolutionary time points at which genome has changed. So, we are going to look into some of these issues. That field is called as comparative genomics, wherein we are going to compare the genome of various species and then try to infer as to what possibly could have changed and how that change has really resulted in a species which is very successful in its own environment. So, these are the issues that we are going to look at it. So, let us first look into the difference between the genomics that we have looked at as compared to what you call now as a comparative genomics. In genomics, normally what you look at is that you are looking at one gene, its function at the various stage or several genes of a given species and compared against all other known genes, try to understand how the gene has evolved and so on. In comparative genomics, what normally is done is that the entire genome of a given species is compared with the entire genome of a large number of species wherever you have the genome sequence. That would give you an idea as to how the genome per se has evolved. In genomics, you use genomic knowledge to understand the organism, so how successful the organism is and how that gene contribute to that fitness. So, here in comparative genomics, normally you try to use the information from many genomes to learn more about the individual genes, how a given gene has changed over time, therefore, the function of the gene is altered in different species. A give example is some of the venom, the toxin that is produced by many of the reptiles and other species are nothing but a very similar kind of protein that your skin makes, your intestine makes. But here this particular protein is there to kill the microbe that enters your digestive system or that try to grow in your skin. But this has specialized in certain species, there has become a venom which is able to kill even much larger organisms, including human. If you look into, for example, snakes, that they have venom that could be neurotoxic and so on. So, it is the same set of gene, depending on the species there were some alteration that led to a specialized function. So, these are the information that you are able to get by looking into the comparative genome aspects. So, what are the benefits? Let us first look into some of the well-known established benefits of this field or emerging field called comparative genomics that would give you an idea as to how powerful the tool is in sort of comparing the genome, what kind of information otherwise you would not have gotten by looking at simply a genome of a given individual or given species. So, this would help us to tell us how did our genome evolve. So, we talk about humans, Homo sapiens, we are all very different from, for example, primates, the apes. We believe that we evolved from a common ancestor for us and the apes, but why we become so successful as a species that we are able to rule this planet, whereas the primates still remains primates, right? And we will give you one example as to how a change in the genome, a particular sequence, probably altered the way this, your skull can hold much larger brain that probably helped in evolution of the human or Homo sapiens. And likewise the language, for example, you are able to speak, right? This, the ability to speak is because of the way your voice box is organized, the way the muscle is there. So, you can imitate a crow, you can imitate For example, many of the birds and animals, you can produce sounds, we can use language to communicate. This again is because of some changes that happened in the genome, which led to the formation of certain different kind of proteins, which helped us to make this sound. And then of course you have the ability to develop a language, process it, analyze, and and develop. So that it two-way process. One, you have a better cognitive abilities. Two, you are able to produce sounds. So these are the two things. What differentiates species? We talk about species. The species is, we always say that species X and species Y had, common ancestor. So that means they all, both of them evolved from a common ancestor. With time, they become two different species. So how really the changes happen? That really help us to understand. And which non-coding regions are important? See, more often when you have discussed in the previous lectures, we are talking about the great majority of our genome are transcribed but not coding any protein. So the transcripts, what you call as non-coding transcripts, have very, very critical functions. This is one, and still we don't know what are the functions. And two, there are regions within the genome which represent control elements. For example, there are some proteins come and bind to, either to activate a given gene or inactivate a gene, or to modify the chromatin the way it should function. So these are the elements that are hidden. It would be very difficult for you to understand where is the element and what is the function. One way to look at is to compare the genome, and if there are regions that are very critical for function, they will not change much. Therefore, looking at regions that are not so diverse, that are conserved across species, will help you to understand regulatory elements. We'll have some examples for that. And which genes are required for the organism to survive in certain environment. This is another important aspects. For example, we have of course the human population spread across the different continents. Some of them are living in very high altitude, some of them living close to the seashore, some are in tropical regions, some are in desert, but still it is not as harsh because we are able to change our environment, right? So when it is cold, you are able to put on some pullover and keep you warm, or make little huts and protect you from the environment and so on. But the animals are not like that. They live in the wild, and still some of them are extremely successful. For example, there are fish that live in the deep ocean where there is no light, but there is tremendous pressure from the top. So the bodies are adjusted such a way and they are able to produce lights. And likewise, there are species that live in desert and in conditions that are hypoxic and things like that. We will discuss some of those issues as to one model animal which really can help us. There are animals that don't get cancer, no matter what kind of mutation you try to produce, they never get cancer. So the genome has the ability to withstand any such damage and produce, or get rid of the cells that can produce cancer, and they don't age. I'll give an example of one such animal. There is absolutely no aging process. They live beautifully as young, and they have all the resistance that you talk about. They can live in extreme conditions. If you can understand what are the genes that really help them to survive, possibly we can learn from them. So these are some of the examples. We'll go more detail as to some other aspects, that is some outcomes of comparative genomics. I am going to give some 5-6 examples before we get into the way we study comparative genomics. So what we say is 60% of the genes are conserved between drosophila and humans. This essentially means that the two organisms appear to share a core set of genes. Now, if that is the case, we can say that 2/3 of the human genes known to be involved in cancer have counterparts in the fruit fly. What does it mean? So you can use the drosophila as a model to understand how genes that are conserved between human and fly are involved in the process of the tumorigenesis, right? There are some, I'm giving you some examples how people have used it. It's separated by 600 million years. That's the evolution that we are talking about, fly and the human. But still there are large number of genes where you could really look at several phenotypes. If you lose a particular gene, what is the kind of phenotype that you can see in fly, and what is the phenotype that you see in the human. There are many that show extremely high conservation. That is what is shown here. Now you can see here, for example, wherever you have this green color arrow bars that represent one gene in fly and one human homolog, and then what happens to such conditions. So there are genes that what you call as essential genes. If you delete the gene, then the fly don't develop, and same thing happens in case of human. The stillbirth or the embryo do not survive, and there are problems what is called a rare congenital problem. There are developmental anomalies. There are number of examples, and there are examples wherein you have one gene in drosophila, but there are more than one gene, very similar genes in the human. Probably it evolved what is called as duplication process. It's because the genome has undergone a lot of changes. We will discuss some of those issues. In this process, the gene number would have increased, made multiple copies, and each one slightly differentiated from the other, but they have overlapping function. But if you have 3 such copies of all 3 are mutated, you may have a cancer. So that's one of the common examples, and so on. So there are a number of genes that shows that kind of a similarity, which really helps us to understand how the genome functions in a larger context. And you can see it even in case of not necessarily only in development, but you can see, for example, there are neurological disorders, there are cardiac disease, there are gastrointestinal disease, and then you can see that all these happening even in case of fly. You can compare the genome functions very similarly. So this is one of the ways by which people are trying to use the information that we have obtained from the fly to understand the human genome and its function in normal development and in pathologies. So this is something that I already informed to you, that you have the process of what is called as reverse genetics and forward genetics. Reverse genetics is that you take a gene, delete it or mutate it and see what happens to the phenotype. And you have a large number of papers and observations with regard to how deleting a gene in fly results in a phenotype or changes in the cellular mechanisms. Now whatever you have gained from this kind of study, you can apply this knowledge to the human, because you cannot do that kind of studies using human population. So you have to infer from what had happened by one way or the other in mutations in the families. But this information that you gain from fly can be applied to the human, and you pretty much sort of figure it out what could possibly the function of this gene in humans. And if, for example, in a fly, if you delete a given gene or overexpress a given gene in a tissue, if it develops tumor, now you can ask the question, if similar tumors in the human, does it have a role for the homologous gene in the human? So you can go and straight away look into whether that particular gene number has gone up, copy number is increased or is mutated and so on. That is one important contribution. The other one that I have discussed in one of the previous class is to model the fly depending on the genetic changes that happened in a tumorous condition or a cancerous condition of a patient. So you have a patient, as we explained, that every cancer patient is unique in terms of what are the genes that are modified resulting in the cancer. So you need to have a drug which is could be more appropriate for that individual. So now you can model that in the fly. You can create all these similar changes in the fly, and then it would develop the tumor, and then you can quickly screen for the drug in a matter of 40 days, 50 days. You should be able to see whether which drug works very good, and then go and use the same drug, because that gives you the speed at which you can create such models, test the drug and come back and try patients. Because you don't want to really waste time in patient by giving one drug and other, because it's going to be normally the chemotherapy and other takes long time, and before you understand that this particular combination of drug works or doesn't work, then you lose time, because the tumor grows and the chemotherapy itself is very toxic to the patient and so on. So fly really helps. So that's where the power of such kind of comparative genomics helps us. Other example is that this approach has helped to find genes that increase muscling in cattle by twofold. It's very interesting phenomena, then, because there are variants in the genome which increases the muscle mass. Okay, and what is interesting is this is not such kind of a variation resulting in the increased muscle mass is not restricted to the cattle. That's what first they were identified, but you can see it in human, you can see it in dog and many other animals. That could have help in understanding the pathophysiology, also to make our domesticated animals much more efficient, right. This is one example I am going to talk about, is that you can see this this cattle is really muscular, and it is because of a gene, myostatin gene is mutated. What happens is this gene or its protein, it sort of negatively regulates the muscle growth. And if the gene is mutated, and then what happens, it does not restrict the muscle growth. As a result, the animal becomes extremely muscular. So they have, of course, because of muscle, they have more power, which could possibly help us, but that is how it was identified to begin with. But it is not restricted to the cattle. Now we have a large number of breeds of dogs also have similar gene mutation. What is shown on the left side is 3 different strains of dog, and these are all wild type as is shown here. So you have the same what you call as myostatin gene. One of the allele is mutated, what is called as heterozygous, or both alleles are mutated homozygous, and you can see that the same animal, if both copies are deleted, then you will find a dog which is extremely muscular, right. And it is true for this species, this is true for this species. So there are dogs are used in different capacity, either for example in the Western world they use it for in the farmland to take care of the territory, to help in gathering the grazing animals, and there are many applications. In snowfield they use it for pulling the sledge and things like that. If there are dogs that are extremely powerful, it's going to help. So there are dogs that can run faster because of the muscle mass. So that is a you can develop breeds, and this is not unusual, because whatever you see, the different breeds of dogs that have long fur, dogs that have long ears, dogs that are lengthy but not tall, all these are nothing but changes in the genome that result to the phenotype. So it's very common in dog breeding to identify a phenotype that is very different from the wild type, to selectively breed them, and therefore it gives you a phenotype which can be used, that animal can be used as a pet or in different context, be it in the farmland or for the police and so on. So that really helps, right. And what really happened to the gene, this is what it is, this particular dog sequence that you can see here. This is just to give you a glimpse of how the automated DNA sequencer electrophotogram looks like. This is what you see here is the readouts of the automated DNA sequencer, and these are the bases that are called, and these are the triplets codon coding for the amino acid, right. This is a wild type. What you are seeing is a wild type. You can see this is the wild type, this is a heterozygous, this is a homozygous, right. And you can see that in the heterozygous condition, from here you find there are 2 alleles coming up. So one that is having identical to what you see it in the wild type, that is TG, AA, TT and so on. But whereas here the lower one represent a deletion. Actually this T and G base is deleted. As a result, there is a frame shift, and you have AATT that should have started here has come in. So this is a heterozygous, and obviously there is a homozygous. Both alleles carry this mutation. As a result, there is a shift in the reading frame, and you have a stop codon instead of the amino acid cysteine, right, which clearly tells you how by looking at several species and comparing the genome, we can identify a gene that gives you the phenotype, right. So this again is not restricted to the dog or cattle. This is also the same mutation. This is the first seminal paper I am showing. Of late there are many other papers. There are rare conditions even in the human wherein you have such conditions. So same myostatin mutation, loss of function mutation results in gross muscle hypertrophy. This is excessive growth of the muscle in a child. You can see that this is a neonate, meaning newborn baby, and this is 7-month-old a baby, but you can look at the muscle mass. They are extremely grown up kind. Look, that's what it is, and it runs in the family. You can see that it is a dominant disorder that from father it goes on and so on, and of course these are conditions which really make an individual powerful. So that's what there is even editorial commentary in the journal which talks about what is called as the powerful genes, right. So here it is completely loss of function, but it's possible that in some of us there could be variation that alters to some extent the protein function. It is not as efficient as it is there in other person. Therefore, you could have really gained lot more muscle mass than a normal person. That could be an advantage for me, depending on what kind of profession I have. It could help, and if it is selected, if there is no other ill effect, then probably it would help. So this is example of that, and there are if you can look into you can Google search, you will find many such babies. One of them I just showing, super strong kids may hold genetic secret, and this kid who is just 3 years old, he is deficient for the myostatin deficiency, but he has a increased muscle mass, therefore he shows a lot more, is more powerful than his peers. That really makes it interesting for at least the kids that are around that guy. It has also important clinical significance. For example, what we know is that this protein myostatin, you lose it, then you put on large muscle mass, right. So if you know how this protein functions in the cell, right, and if you are able to identify certain molecule that can partially block the function of this protein myostatin or its pathway where it is working on, then you can use that as intervention strategy to increase the muscle mass. So this is one paper which talks about such kind of application, at least using cell models, how possibly this can be achieved, and this is engineering. Now I'm going to go one more example and show it's a recent example, happened just a couple of months back in China. You remember we discussed a condition wherein you can the CRISPR-Cas9 model wherein you can edit the genome. Remember, like we said that you can use a guide RNA. With that you have this endonuclease which can go and cut and edit, and then make whatever changes you want. A group in China, what they have done is a normal breed of dog they have taken, right, the cells, embryonic stem cells. They edited the gene myostatin in that embryonic stem cell such that now it has, they have introduced the mutation, and they made a chimera and they produced a dog which is like the hero now, is a muscular dog you can create. So you can create, for example, a breed, a given breed, which is, for example, which is extremely good in detecting, for example, so there are different breeds of dog which have unique capacity. For example, smell, you must have seen the police use the dog to sniff around and find is there any explosives, right. They train, but it's not that any dog strain can be trained for that, because there are certain phenotypes that are selected, and these breeds are very good with smell, right, and they can be trained. And but if assume a dog strain is there, it is able to sniff well, but it cannot run, right. So therefore, this dog can sniff somebody is having an explosive. If that guy runs away with that explosive, then the dog cannot chase. On the other hand, if you can make that particular dog to run faster by putting more muscle, then you are able to create a new breed which has the desirable characteristic, that is, you are able to smell whatever explosive in others. At the same time, you make them to run faster, make it stronger. So that is an advantage. So these are some of the applications now people are trying to introduce in domesticated animals, so which possibly could have some desirable effect. So this is how the comparative genomics really helps. The other example, such approach also helped us to map genes that increase the yield of high fat milk in cows. So it increases because milk is one of the essential commodities really all of us need, and if you can produce cows that have a high fat content milk, then possibly you could be of great help. So it results in higher production level and potentially significant economic impact. It's going to really help. So again, people have used genomic approach. People have sequenced the different groups have sequenced the genome of various cow species that produces a either high, for example, volume of milk or high fat content. And if you can bring these together, obviously it's going to be much desirable end result, because you're going to have more milk with high fat content. Is really helpful. And there are approaches people have used to identify, for example, the gene which impact, which help in the high yield, right? And then now they have identified these genes. Now if you can likewise manipulate or engineer the genome, probably you would be able to have cow breeds that could really produce large volume of milk. Another example is how even birds can help us to understand how we are able to speak, right? This is a landmark study again. Comparisons of nearly 50 bird species genomes revealed a gene network that underlies singing in birds that may have an important role in human speech and language. So the language is not limited to humans. There are all the species communicate. They communicate in different ways. We don't understand always. But one of the fascinating research field that had come from the birds is this birds sing, right? And this is the way they communicate. They have a variety of expressions that really tell whether they are happy, they're predators, they communicate with each other. And it's so for most of the animals, but bird is something which is unique in that, because they have much more sophistication the way they communicate with each other. And that is how they have try to understand as to how what are the genes that help them to make these kind of songs which convey certain information, and how the brain is able to process the information and store and to get the information out of it, because there also has to be a memory, because it's not one time event. So how does the brain is able to receive this information, store it and process, and then get the meaning out of it. This paper came in Neuron in year 2012, where they have really looked into a large number of genes and the networks, how different genes function together, and that is what called as molecular microcircuitry, and that pretty much it helps you to understand how at the neuron level the information is processed and stored, right? So that's kind of learning ability. And then once these birds, the young ones, learn from their adults, then they are able to practice it and give it to the next generation and so on. So that's the way they communicate with each other. And this is not only that whatever you understood only birds, but the processing is also the understanding how the brain processes the information might help us even humans, because there are kids who have inability, meaning they are challenged to speak or to comprehend what people are saying. So these are called as speech disorders. Either they are unable to put together words and communicate, or they are unable to understand what has been told, right? So this is because the way the brain is able to process information, somewhere there is a problem, right? So what goes wrong in these kids is going to be very extremely difficult to understand, but the birds might, the study from the birds might help, because what we know now is that the song circuits, right? So what has been compared here is the kind of human brain, mammalian brain with the bird brain and so on. And what we are saying is the way the bird's brain process this information seems to be very similar to how the human brain process information, especially the language. So if you can gain deeper understanding as to what are the genes that are involved in this, possibly you can go back and look into human population wherein you have the speech disorders, and then see whether these genes have any changes that possibly explain why you have this disadvantage. So you can see again how studies from one species can really help us to understand the other species, including human. Again, example of dog. Here as I told you, there are huge number of breeds available for dogs. There are many dogs that also have cancers, right? either they are more prone for cancer or they are very good model for cancer, and spontaneously they develop cancer. So that's since they are being higher mammals, you can study them, and that would help us to understand the genetics of cancers in human as well. And there is again a paper which talks about how the dog models of naturally occurring cancer, these are not induced cancers, these are naturally occurring in the dog models, and how this study can help us to understand the genetic defects behind such cancer form. And one way is to look at, for example, the gene c-Met, is one of the genes, the oncogene, which when it's activated, then it leads to cancer. We can study that in, for example, in dog, or there are breed specific germline variants. There are variants that happen, variation that happens in the genome, which makes the animal more at higher risk of developing cancer later, certain breeds, right. That then one can go back and look into what are the variations that are there in the dog genome that made it at a higher risk of developing cancer. And then we can go and compare, for example, with other species, human and other animals. That will help us to understand the genomic signature that can predispose to a given cancer. And like what you see in human, there are somatic alterations, meaning only in certain cell of a given tissue you have changes in the DNA which results in cancer. So we can do that kind of a high throughput genomic analysis in cells that represent cancerous state and compare it with cells that are normal and try to understand what genomic alterations happen. And you can, in fact, he can correlate with the humans as well, which gives you insight as to which possible region in the human can have very similar effect if altered, right. So there are huge number of advantage, including, for example, genomic instability. There are regions that are duplicated, meaning 2 copies, or there are regions that are deleted. Any one of them, copy number variation again is one of the major triggers for cancer, and then there are dog models which really help us to understand that. So this is again a very, very powerful model system to understand. If you know the genome, you can do. And there are also examples of what is called clinical relevance of dog for cancer treatment, because they spontaneously develop cancers. Now we can try out different chemicals, therapeutics, to see how effective these chemicals are in controlling the cancer. So you can see here, this is one that is what is called as treated with a given drug, and what is shown here is a survival, right. Then percent animals survived over the time, and this line represent the one that is treated with vehicle, meaning untreated. And you can see that over the time you will find that in about 7 weeks, almost 70% of the dogs have already that had cancer already died down, whereas the one that is treated, they are able to live longer, which gives you very good model to even for drug screening platform and so on. So this is another implications, because what is the genomic changes happened in this particular cancer in these dogs, and you are trying to use a drug. So once you know that the drug is able to control the tumor growth in this model, then this can be easily applied to human, those who have got same kind of alteration. So that really helps us to model cancer as well. The other example is you can also understand how genes affect body shape, size. Again, dogs are good example, and how really it affects to better understand growth and development. You have to you can relate everything. For example, your skull size, right? It's very important. The face features and your ratio between the skull to the body and so on. There are many constraints, because if that constraints are altered, then it's going to have a problem even in birth, because if the baby's skull size is much larger than desired, then there cannot be a normal birth, because the baby has to come through the pelvic bone, and there is a small window for the baby to come through. So it's going to be a limitation. So there are strict control over the genes and how they regulate the skull function. And this is one example as to how there are different breeds of dog which have got different shapes of skull. again that each one can be linked to a particular gene signature, and that is what is shown here. There are you can look into, and people have mapped it. These are regions, for example, chromosome 1, chromosome 5, chromosome 9, and so on. There are different regions, different chromosomes. People have mapped genes that regulate the skull size and skull length and the complexity, right? Which again is a very good way of understanding the gene and what are the set of the genes that are involved in the governing the body shape, right? Now what you do is you basically sequence a particular species of a dog and compare with the genome sequence of another dog. These two have difference the way the skull structure is. Then you look into what are the genes that are different and try to understand their function, and which would help us to map the gene for this particular phenotype, right. So this is again a powerful tool which really helped us. So the man's best friend becomes biology's best in show, meaning you can look at if the dogs have been domesticated a long time back, and they have been living with the man. They call it his best friend because they always help. Unlike other species, like whether it is a pig or cow or cattle or horse, they have a specific function, right? Mostly horse for transport. You have a pig mostly for food, and likewise chick and so on, or cattle for either workhorse or for the milk. But dogs have variety of utility for the human, right from being a good pet, right, to being a ferocious defender of your land or your house, to in snowfields, for example, they can sledge, for example, they pull the cart, and there are variety of help that the dog breed gives. As a result, for each one of the specific functions, people have over the time they've selected breeds, identified them, they bred, and they have pure inbred strains which have a definite phenotype. So this variation in the dog breed, because of the selective breeding, encompasses a large range of size, shapes, color, and behavior. As I told you, the behavior, sniffing around, because they have been selected for this particular quality over and again by breeding, selectively breeding. So they have the extremely versatile way of identifying different smell. You must have seen in movies, where there is a victim who is dead, and you want to see, and there is in the crime scene there is a cloth that are left, not probably belong to the victim, but it could be because of the person who possibly killed him. And then they show the cloth piece to the dog, and dog goes around and find the person who culprit who killed this person. So this you must have seen, but it is because they are able to distinguish the smell, and they have a beautiful well developed odorant receptor which is able to distinguish various smell types and able to map them as well. So that is an important aspects in dog which really help. That's why the dog species have been extensively studied, and it has led to a large number of discoveries which could have a implication even in the human. For example, one of the landmark study identified a sleep disorder, and this disorder affects the dog, and later the same gene mutation is known to cause the same disorder in the human. So really it helped. The other example is another dog model developed a very fatal form of epilepsy because of a genetic change that is there, and the same epilepsy caused by the same gene mutation is known in the human, right. Now in addition to understanding the conserved function of the genes and the mutations process, these dog model really help us to even understand the pathophysiology. So that way the dog has been one of the best studied example, and really helps us to understand various process of genome evolution, and that's pretty much brings the end of this particular lecture, and we'll see again in the next lecture.